My name is Medina. I'm the co-founder and CEO of Nci Labs. We are Nci Labs and we're going to help translate nanomedicines by providing AI predictions of nanoparticle interactions with biological systems. What are nanomedicines? Well, think of it as a spherical system made up of two essential parts: a container that is used as a delivery tool and a cargo carried by this container. This container can be made up of different materials, such as inorganic nanoparticles like gold, or biological materials like lipid or cholesterol, with different structural, mechanical, magnetic, electrical, or biological properties. And your cargo can be anything, ranging from vaccines to anti-cancer drugs. Nanomedicine's field has an enormous potential, but designing novel particle systems is the least challenging part. You need to be able to deliver them to your targets. Well, one way is to deliver your nanoparticles passively. Imagine you are holding a helium-filled balloon in your room and you let it go. Now you have no control over where it will end up. Another way is to deliver your nanoparticles actively. Active targeting relies on the ligand-receptor binding. Now let's say you have a particular spot on your ceiling where you want your balloon to end up, and you tag that spot and you tag your balloon with sticky tapes and you let it go. Now you have control over where predominantly your balloon will end up. In the last 20 years, only few passively targeted nanoparticles have made it to FDA approval. Successful translation of actively targeted nanoparticles into clinics remains challenging. There is no existing actively targeted nanomedicines in the market. This is because once inside biological fluid, nanoparticles form a new biological identity. It becomes larger in size, and its surface targeting ligands are now shielded by biological molecules. In other words, the sticky tapes on your balloon are now covered and not available. Which brings us to the second problem. The size of your nanoparticles has increased, meaning they cannot pass every biological barrier that you initially planned for them. Since your targeting ligands on your particles have now been shielded, you are now left with passive nanoparticles that does not exhibit preferential binding to your target receptors. Additionally, biological identity of your nanoparticles and consequent of target toxicity is individual and disease specific. Our company is developing a software to predict, depending on nanoparticle size, type, surface chemistry, nanoparticle interactions in different protein environments. Using NanoAssyst, you will be able to model your nanoparticles in vivo environment, understand nanoparticle interactions with biological systems and come up with new strategies, understand individual and disease-specific nanoparticle interactions of your actively targeted nanoparticles. Using NanoAssyst, you will design efficient actively targeted nanoparticles, which will decrease the clinical trial failure rate and save billions of dollars. We have a preliminary data and a proof of concept. We are currently working on acquiring more data to validate our deep learning model. And for this, we aim to raise $250K. In our founding team, we have passion and expertise to help overcome the challenges of actively targeted nanoparticles. I'm a scientist working in the field of nanomedicine for the last 7 years, and my co-founder Riyad is a deep learning expert with experience working for machine learning startups. We are supported by advisors from NPL, UCL, British Society for Nanomedicine, and NHS. Thank you for listening. I'm grateful to the team behind programs that Nci Labs has been part of, including UCL Bloomsbury Academy, P4 Precision Medicine Accelerator, and Conception X. If you have any questions, please feel free to send an email to info@ncilabs.co.uk.